Hello, Stephanie.
Hello, Joanna.
So good to have you here with us. So this is our third Greener Talk organized by Axia Innovation in the context of the Horizon 2020 Greener project. And this time with me is Dr. Stephanie Roosa from Materia Nova, a member of the Greener Consortium. Stephanie, would you like to briefly present yourself?
Yes, sure. Thank you for the kind introduction, Joanna. So as you said, I'm working in Materia Nova, which is a research center in Belgium. Our aim is to perform applied research about innovative material and processes. And I am program manager for the biotechnology department. My background is environmental microbiology, and the specialty of our lab is the use of microorganisms. We call that, it's called biocatalysis, for diverse applications. It can be the production of a specific molecule, the degradation of components such as plastic, or for depollution purposes, which is the aim of Greener.
Great, thank you. So let's start with our first question related to the involvement of Materia Nova in the project.
Um, so in the Greener project, let me specify that we are two teams from Materia Nova involved. So the biotech department I just mentioned, and my colleague from the LCC department, which are involved in the impact evaluation of Work Package 7. But today I will talk mostly about my field of expertise and our contribution as the biotechnology team is mostly related to Work Package 4, so the development of water remediation technologies, and Work Package 6 for its upscaling. And we also contribute in Work Package 3 about the characterization of water sample, the isolation of metal-resistant bacteria that we are using in Work Package 4, and also a small contribution in Work Package 5, for which we produce a specific enzyme for soil depollution. Just to give you more explanation about our process, in the process, in the Greener process we develop, we use the ability of bacteria developed for to fight against metals. So bacteria in general have developed different resistance mechanisms, either to mobilize or immobilize metals. And in the framework of Greener, we are focusing on the immobilization of metals as nanoparticles. So there are different species of bacteria able to do that. I've added some picture of the bacteria we used in Greener. As you can see, some are producing chains of iron particles. Others are coating almost completely with gold nanoparticles, among other particles. And there are even others producing really, really small copper particles extracellularly. So all these bacteria have their specificity about the production of particles, and also, so the composition, the size, for example, and also about the condition they can survive to. And as we know, environmental contamination is not simple at all. It's usually very complex. It's not only about one metal, but multiple metal at various concentration, and often other contaminants are also present. So our strategy in Greener was to combine all these bacteria, the bacteria we've selected as consortium, in order to, well, first address the complexity of environmental pollutions, and also to produce different types of particles. So technically, we selected 5 bacterial strains for their ability to produce particles and also to survive under high metal concentration. We tested on a Greener sample from a very contaminated site. the water was very challenging because the pH is very acidic. The copper and iron concentration were very high in terms of gram per liter, and there is also nickel, arsine, cobalt, lead, really complex matrix. So we tested the bacteria alone and then assemble it as a consortium, a different consortium. Uh, and then it enables us to select one specific consortium based on two bacterial strains, based on their efficiency also to remove metals, but also to produce nanoparticles. This two-strain consortium has been proven now to be quite effective at lowering the metal concentration. We reach between 10 and 25% of copper and iron removal within 1 day. And we also managed to recover some particles. As you can see on the picture, these are the magnetic particles that we recovered. And then now we are optimizing the process and continuing the upscaling from a really small flask to a 10-liter reactor.
Great, thank you very much. This is actually very interesting and a very challenging approach and the work done under Greener. And as I understand, this is a collaborative work, right?
Yes, indeed. To be honest, to my point of view, collaboration in general is key in research projects because it enables to share ideas, expertise, and that's also what makes such project very interesting. And more specifically for the process I just explained, we collaborated with other partners among the consortium, mostly to integrate this process into as a hybrid technology. So these bacteria are now used by the University of Surrey to be integrated in BES, and also by the Joseph Stephen Institute to construct artificial aggregates. And then we also work with Tau for the development of a tool to quantify specific bacteria in a bioremediation process. But of course, we are interacting with other partners to organize the projects and for other aspects also.
And lastly, could you briefly present the benefits that EU can have from the bioremediation technologies that are developed under Greener?
Yes, sure. So the technologies developed in Greener are answering the European needs about the water reuse, the accessibility to water, and to build a healthier planet, which is described, for example, in the Green Deal. And the legislation are slowly but surely changing to frame this. So the rules will become more and more drastic about the quality of water, about the quality of industrial water released in the environment. And in view of this, then the greener technologies are therefore helping to meet these requirements. And that was for bioremediation in general, but more specifically about the process we developed. You know that the nanoparticles are used in many objects of our day-to-day life. So there is an important need of these nanoparticles. And to obtain it, Europe is largely dependent on other countries to import these nanoparticles. So it would be ideal to have find a solution to lower our dependency and produce it from Europe. And so that's the reason why we propose to produce metallic nanoparticle and from contaminated water. So it's therefore an environmentally friendly strategy to depollute waters while obtaining products with an added value.
Yes, I was saying that that's very well said. And yes, I think that this project has a lot of potential, both in the EU and globally, for the solutions that it is proposing. So, yes, with this being said, I want to thank you for your time. And yeah, we will see you on the next Greener Talk. Thanks again.
Thank you very much, Joanna.